Okay, hi everybody. This is another joint session of the Martinos Center BrainMap Conference and the MGHA Taxi Center Noon Conference. And the reason for the special conference today is that we are very honored to have with us a friend and colleague, Professor Chris de Zeeuw from Rotterdam. Chris is actually, we decided that we are related because Chris's institute that he is the head of at the Department of Neuroscience at Erasmus was founded by Hans Kuipers. And Hans Kuipers was then followed by Dr. Jan Vught and then Chris. And Hans Kuipers was a teacher of my teacher, Dr. Deepak Pandya, from the early papers of Pandya and Kuipers in 1969. So we are, we're I think that's what, second cousins, I think that's how it works, something like that. In addition to that, Dr. de Zeeuw is one of the world-renowned physiologists in cerebellar neurology, looking at underpinnings of the cerebellar contribution to movement and now to cognition, and his elucidation of the principles and organization of cerebellar neuronal architecture and physiology has been very powerful in helping drive the field forward and understanding how the system works. So we are very glad and honored and privileged to have you join us today. So Chris will talk about cerebellar orchestration of motor behavior and cognition. Thank you. Welcome.
Thank you very much, Jeremy. Thank you very much for organizing this lecture at the very last meeting, at the very last minute. I'm happy you're all here. Please feel free to interrupt me whenever you want. We're going to talk about very fundamental mechanisms of how the cerebellum is working. And then I will take it actually to topics that I think are relevant for the people here, the role of the cerebellum in cognition, as well as some clinical relevance. So I will start off with some fundamental principles, and then gradually we'll move on to the topics that are being investigated right here in this institute. So if you think about the cerebellum, how it is organized and situated in the brain, you can make this kind of scheme, right? So we have the cerebellum consisting of the cerebellar cortex and the cerebellar nuclei, and they have impact on the brainstem, yeah? And from the brainstem, they can either directly control motor behavior or they can project to the cerebral cortex, where they can, for example, also influence cognitive processes. If you think about the organization within the cerebellum itself, there are two main inputs. One is from the mossifibers, largely derived from the pontine nuclei, but not exclusively. There are other nuclei in the brainstem, tens of smaller nuclei that also project mossifibers to the cerebellar cortex, as well as to the nuclei. And on top of that, we have the so-called climbing fibers, indicated here with CFs, and they are all derived from the inferior olive, so exclusively from the inferior olive. And they are important, as I will try to convince you today, for the orchestration of cerebellar activity. And so you can see this also when you look at the map generated by Jan Vogt, my predecessor. If you make a map of the cerebellar cortex, you can divide the whole cerebellar cortex in these so-called sagittal zones. Yeah, and you have the mid part, which is controlling motor domains of the trunk muscles. Then when you go more lateral, you will have motor domains controlling the arms and the limbs. And here, for example, when you go to the pink area, this is the vestibular cerebellum. There, for example, eye movements are controlled. And on top of that, again, more lateral in the cerebellar cortex, you have these areas that are sort of a bit unclear yet, which are probably involved in cognitive processes. So we will come back to that later. Now, already, let's say over the last 50 years, many molecules have been discovered that are distributed in a kind of sagittal fashion. So here you see the labeling of aldehyde, also called Zerbin-2. And you see in the whole brain preparation, you see of a rodent, you see here these sagittal stripes. And so it's very important to understand when we have a coordinated behavior, like when I use my arms and while I'm walking, all these different sagittal zones can work together, and they work in a sort of coordinated fashion. And this is most likely being controlled by these main efferents, particularly those of the inferior olive. Let's take a little look here how widespread these zones are. This is again the labeling, the presence of LDLC or zebrin. And when you rotate the cerebellum, in this case of a mouse, you see the striping pattern, how prominent it really is. So we have taken this and investigated further at the molecular level together with others. And what you see is if you look at the transcriptomics of all the genes expressed in the different zones, you see that there's a real organization. So for example, if you take all the genes that are expressed together with LDLC, yeah, you really have tens of genes that are expressed together in one particular zone, in this case, particularly in the zerbin positive zones. And then you have other zones, which we call zerbin negative zones. They are sort of always expressed in PLC beta 4, and they have another pattern of genes. So they are largely segregated, pointing towards a dichotomy also in the function of cerebellar cells. And these genes are largely related to what's expressed in the Purkinje cells themselves. The Purkinje cells, they form the major output neuron of the cerebellar cortex, and they project to the cerebellum nuclei. Now, when we saw all these genes passing by over the years, we had the idea that there must be a functional correlate. So we started to record the simple spikes and the complex spikes just randomly from the cerebellar cortex. Yeah, you see in blue the so-called simple spikes. They can fire between 50 to 100 hertz during baseline activity. And then on top of that, you have the so-called complex spikes, which only fire under normal circumstances like 1 hertz. The complex spikes are triggered by the climbing fibers. The simple spikes are intrinsically generated by the Purkinje cells, but can be modulated by the multi-fiber inputs. And when we start to randomly record in the whole area of the cerebellum, in the anterior lobe, in the posterior lobe, and within the same lobules from zerbin positive going to zerbin negative zones, yeah, you could do this in vivo with the E84 GFP mouse. And we found that the simple spike firing frequency in the zerbin negative zones is significantly higher than those in the zerbin positive zone. So in the zerbin negative zones, it's about 90 to 100 hertz on average baseline. And in the zerbin positive zones, it's more like 50 hertz. So here again, you see the difference in firing rate in climbing fiber pulse that occurs after the complex spikes is shorter in the zerbin negative zone because of the higher firing frequency of the simple spikes as compared to the zerbin positive zones here. Now, when you look at the cytoarchitecture, how is the cerebellar cortex organized? Here in blue, again, zerbin positive zone, in red, zerbin negative zone. You see that they're virtually identical. There's only one difference, and that is that in the zerbin positive zones, we have the so-called unipolar brush cell, the UBC. Now, they have a particular function that is still uncovered. I can tell you more about it, but I will not do that today. I will do that at the Society for Neuroscience. But other than that, they are actually exactly the same. And because the intrinsic firing frequency of simple spikes of the Purkinje cells is lower in the zerbin positive zones, we had the hypothesis that during learning, because the initial firing frequency is relatively low, that during learning, the activity would go up. And therefore, we call this an upbound module. Yeah, and vice versa, when the simple spike firing frequency is already high, there's ample room to bring it down. Therefore, we call it downbound module. And this was initially a hypothesis, and I will present you some data today that actually support this hypothesis. So here's the idea. If you look at the initial firing frequency in the zerbin positive zone, here simple spikes are low compared to the high simple spike firing frequency in the downbound module. And then during learning, the firing frequency would go up in the upbound module and down in the downbound module. The inhibitory molecular interneurons, they should show the opposite pattern because they are inhibitory largely. And so if the simple spike go up, the molecular interneurons here have to go down in the zerbin positive or upbound module, and vice versa in the downbound module. Now, all these kind of activity changes, we hypothesize, are mediated by activity of the complex spikes. They can have impact on all the forms of plasticity in the cerebellar cortex. So the rule is such that when the complex spikes go down, simple spikes should go up, and again, vice versa. And this happens through mechanisms, various mechanisms of plasticity. So, for example, when the complex spike firing frequency goes down, as indicated here, then you will get long-term potentiation of the parallel fiber to Purkinje cell synapse, which will then increase the simple spike activity. And the opposite should happen in the MLIs because they are in the end inhibitory. So when you get LTP at the parallel fiber to Purkinje cell synapse, you'll get depression, long-term depression at the parallel fiber to MLI synapse and at the MLI to PC synapse. Of course, the opposite then will happen here. And so when the complex spikes go up, you get long-term depression at the parallel fiber to Purkinje cell synapse and long-term potentiation at the MLI synapses. And this is not a kind of coincidence, right? When you look at all these studies that support these hypotheses, this is actually showing that the complex spikes, they can really orchestrate all the forms of plasticities at the same time. Yeah, initially it was only thought that complex spikes would only have impact on LTD at the parallel fiber to Purkinje cell synapse. But as the years went by and more studies were done and the molecular mechanisms were uncovered, it really showed that the climbing fibers, even though the firing frequency of the complex spike is very low initially, they can have a tremendous impact on all the forms of plasticities that are occurring in the cerebellar cortex. And they operate in such a way that they work synergistically, supporting the idea that in the end, what you want to do is bring the simple spikes up or down in a concerted action. Now, so this is the first hypothesis is the actual change in the simple spike activity. The second hypothesis is that it comes together with all these different forms of plasticity. And the third hypothesis, which is also really important if you think about it from a neuroanatomical point of view, is that downstream of the Purkinje cells, in the end, what you want to do is change the activity of motor neurons if you want to control sensory motor behavior. But in other cases, you can think about activity changes in the cerebral cortex, like when you want to control cognition. What you in the end want to do is in the upbound zone, as well as actually in the downbound zone, in both cases, you want to activate the motor neurons. Yes. So then taking into consideration that Purkinje cells are inhibitory, yeah, it means that the net polarity of the synapses downstream of the Purkinje cells should be negative in the upbound zones to get a positive effect in the motor neurons. Whereas in the downbound zones, it should be opposite. The net polarity should be positive. Again, yeah. So because when the simple spikes go down here, it means less inhibition, which means more activation of the motor neurons, and vice versa in the upbound zones. Because why otherwise make it so clearly up and down bound if you cannot link it directly to the anatomical connections downstream? So these three hypotheses, I will first try to convince you that they are actually correct by looking at different sensory motor behaviors. This is the first part of my talk. Then I will go into how they may act during cognition. And then at the last part of my talk, I will briefly tell you something about some clinical implications of these general concepts. So let's first look at sensory motor behaviors. We have looked over the past decade at many behaviors, including also whisker movements, locomotion, and others. But for today, given the limited amount of time, I will only talk about two sensory motor behaviors. For the upbound zones, I will talk about adaptation of the vestibulo-ocular reflex. And for the downbound zone, I will start talking about Pavlovian conditioning. So let's start talking about the adaptation of the vestibulo-ocular reflex, which takes place in the flocculus. And as you see, the flocculus is basically entirely Zeeuwian positive, so we expect an upbound phenomenon, an upbound change during learning. Let me first show to you how important this reflex is. Here you actually see the cervical ocular reflex, but look at how stable the eyes of the owl are in space. No matter what the experimenter is doing, both the eyes and the neck are very stable. Even when you close the eyes, the head is stable. Yeah, so it's a very powerful reflex, and we can mimic this in the lab. So here we have a turntable. Actually, this is in my lab in Amsterdam. There's a mouse in the middle, and we have a camera detecting the pupil, and we can measure the movement of the pupil with respect to the corneal reflection of the light. And then we can play with both the movement of the table and the retinal image that's provided by a projection system. So, for example, in this case, the retinal slip is increased because the visual image is rotating in the opposite direction to that of the table rotation. And if you do this for like half an hour, then you put the animal back in the dark, it means that the gain of the eye movements will have been increased. Yeah, you can also do the opposite and bring it down, and you can play with the face and do all sorts of things. But yeah, so let's use this system and see whether the three hypotheses that I posed are actually correct. This is the brainstem system controlling activity in the flocculus that during learning then will have to motivate and increase the gain of the VOR. And we have recorded the activity of the simple spikes while measuring the eye movements. And then indeed what we do see is that the firing frequency of simple spikes increases when we induce this type of learning that I just showed you. Yeah, so you can link directly the output of the behavior to the activity of the neurons in terms of simple spikes. So it confirms that simple spikes are actually increasing during upbound learning. Now you can say, well, maybe this is an epiphenomenon, maybe this is not really causal, it could be an efferent copy signal or whatever. So what we did is to really prove it, that it is sufficient to induce the learning. We did use some optogenetics. So we inserted proteins into the flocculus of a mouse, yeah, and we were shining light on the flocculus of the mouse with different intensities. And what you could see is that not only did the simple spike increase, but at the same time, the eye movement amplitude was also increasing in a linear fashion in relation to the stimulus intensity and the simple spike rate. So thereby you could show that, well, it is at least sufficient to drive the eye movement response. And we are now making models and so on, and it really looks like this is the causal phenomenon driving the upbound learning phenomenon. Okay, so that was the first hypothesis. The second one was in relation to the forms of plasticity. As I said, we were predicting that to get the increase in simple spikes, you need potentiation of the parallel fiber to Purkinje cell synapse. Yeah, and this is indeed the case. We by accident, in a way, we uncovered that a change in the diameter of the gluate 2,3 diameter MPA receptor subunits, yeah, that is responsible for induction of long-term potentiation at the parallel fiber to Purkinje cell synapse. So this was a completely new hypothesis, because based upon hippocampal studies in the old days, everybody always thought, okay, you have to build a new MPA receptors to get potentiation. Now we uncovered that actually only the receptors that are already there, within the flash of a second, you can induce long-term potentiation. Yeah, this was a completely new phenomenon that later on was also shown in the hippocampus. But here, usually it's opposite. The hippocampus is leading the cerebellar field, but we were instead here leading the hippocampal field by showing that that this phenomenon, which is unique, was driving the simple spikes and thereby also the learning of the eye movement, as you see here. Now, so this was the second hypothesis, that you need LTP rather than LTD, as Ito always proposed. And the strength of this hypothesis was shown in yet another experiment, that if we talk about orchestration by the climbing fibers, how do you show that actually, you need this absence of climbing fiber activity to drive the potentiation of the simple spike? Well, we had an interesting experiment. This is what you see under normal circumstances. So You see, when the simple spikes go down, there's more complex spikes. This is the reciprocity. And when complex spikes are low, the simple spikes are high. And so the eye movement gain is going up in this stage, but down in this stage. So how do you prove that the complex spikes are actually driving this? What we did is we created an animal in which the route of the climbing fibers which are normally exclusively going to the contralateral side. As you see here, these are the climbing fibers from the inferior olive to the contralateral side. We now made a mouse in which the climbing fibers are derived from the ipsilateral side, while the mossy fibers were still unaffected. And so what happened then, indeed, we could show that in these mice, the complex spikes were completely reversed. So instead of, let's say, having an optimal activity here in the middle, the activity went to the other phase of the cycle. And then as a result, the simple spikes also completely reversed. Yeah, so here you see, as compared to the simple spike modulation here, it's completely reversed. So it means that the phase of the simple spikes are not determined by the mossy fibers. They can, in the end, completely dominated by the climbing fibers. So they are the real conductors of the orchestra. And of course, the mossy fibers can set the context, and they can determine whether the amplitude is going to be very high or mediocre high. But the phase, the timing, is really conducted by conveyed by the climbing fibers. And this again shows that how powerful this LTP is, because if you reverse the phase of the climbing fibers, only when the climbing fibers are low, like here in the middle, the simple spikes can go up. This is the stage when LTP is expressed. So it confirms completely the plasticity hypothesis for the upbound zones. Then, of course, we also looked at the net polarity in the postsynaptic synapses of the VOR system, and it was indeed negative, as we already knew from many classical studies by other people looking at the vestibulo-nuclear neurons contacting the motor neurons and so on. This is, of course, a relatively simple schedule, but you can think of much more complicated upbound modules, for example, those involved in locomotion or in cognitive processes. We will see that later. But then again, the same rule holds that whenever you have an upbound zone, the net polarity is negative to get, in the end, the effect that you want. So also the third hypothesis was confirmed. And here you see the three hypotheses together, all working together in the way we predict according to our model. Now, what is going on in the downbound module? The downbound module, for this, again, as I said, we looked at Pavlovian conditioning, delay eyeblink conditioning, and this takes place in various areas, but among which the deep fissure of lobule simplex. And for those who don't know how it actually works, what you do is you have a conditioned stimulus here indicated in purple, and an unconditioned stimulus, which can be an air puff, indicated in blue. And during delay conditioning, the conditioned stimulus, for example, LED light, is continuously applied until the US is terminating. Yeah, and then this interval, which can last a couple of 100 milliseconds, for example, 250 milliseconds, will be given for a couple of 100 trials to the subject. And initially, of course, there's only the unconditioned reflex when you give the unconditioned stimulus, and nothing happens in the interval between the CS and the US. But as days go by, you can see that every single day there's an accumulative effect, and there's a conditioned response in the end, such that the eye is perfectly closed when the puff is coming in. Yeah, so you get this in a nice conditioning of the eyelid response, and if you will not even give the US anymore at the end, the eyes will still close. So you give the CS only response. Okay, so this is the behavior. So how do we test it? First, of course, we just look at the activity of the simple spikes during the conditioning process. And here you see the Purkinje cell activity, and you see indeed that, first of all, that the simple spikes start from a high baseline firing frequency, about 100 hertz, as you expect from a downbound zone. And then indeed they go down during the learning, during the expression of the eyeblink response. So this is at the end of the learning. You see here the conditioned response. This is the unconditioned response. And again, so we see a simple spike suppression as predicted. Now, with regard to the plasticities that are engaged, we expect both LTD to play a role and potentiate LTP of the parallel fiber to Purkinje cell synapse to bring the simple spikes down. But we also expect to have potentiation of the MLIs, both at its input and output. Why? Because Purkinje cells themselves are intrinsically very active. And if you, even in the slice, when you take away all the inputs of the Purkinje cells, they will still fire a large amount of simple spikes. So to bring them actively down, LTD, let's say blocking the parallel fiber to Purkinje cell synapse will not be enough. You need to activate the MLIs to bring it really down. And so we predicted that you need to have a concerted action of manipulation of LTD and LTP of the MLIs. First, I show you that actually, interestingly, that the amount of LTD that can be induced in the downbound zones is significantly higher than those in the Zeeuw positive zones than in the upbound zones. This was done by Stijn Voerman recently. And so as we hoped and predicted, let's say the amount of LTD that can be induced in the downbound zone with the same kind of stimuli is higher than that in the upbound zones. Then we expected that MLI activity would go up, right? So the MLI activity, the molecular layer interneuron activity did indeed go up during the expression of the eyelid response, also in line with our prediction. So we expected potentiation of the MLIs. And this indeed all turned out to be true. We manipulated simultaneously the activity of the MLIs and of LTD in the Purkinje cells, and this was shown by Henk-Jan Boele. Indeed, when you affect them both, only then you will get a virtually complete blockage of the Pavlovian conditioning. So if both don't work anymore, only then you will get a strong phenotype. We published this in Science Advance a few years ago. So again, we can say that the first two hypotheses are confirmed. And when you look at the polarity downstream of the oculomotor of the Purkinje cells, you see that indeed all the connections here are positive, yeah, as indicated here. So also the third hypothesis was all in line with what we expected. Now, this doesn't mean that the whole theory, let's say, of upbound and downbound modules is can be considered as established, because there are I should say there are always exceptions, small exceptions. So for example, in the eyeblink zones, there is a very small mini microzone, you could say, where actually we see the opposite. So that's maybe 10% of the cells, but we still see them, and we're trying to figure out what they do. It could be that they are actually important for stopping the conditioned eyelid movement, those kind of things. But by and large, this model is very solid, I would say. Okay, so for both the upbound and downbound zone, all three hypotheses can be confirmed, I would say, to a large extent. Now, let's try to see how this translates to more cognitive tasks. And for this, I would like to first show you a sort of extension of the eyeblink task, but then in real life, shown by Ronaldo. Let's first look at what happens.
Gives him the ability to intuitively read the game under normal conditions. But what if the conditions are very far from normal? Can Ronaldo still hit the back of the net if all the lights are turned off and he's in complete darkness?
Okay, what we're going to do, we're going to have Andy feeding the balls in as if it's a cross, and then sometime in the ball flight, the lights are going to go black, so you won't be able to see anything at all. We're expecting you to be able to put the ball in the goal. That's what we're hoping will happen. We're hoping that you'll have picked up some advanced cues from Andy's body position, the shapes, the positioning of his feet and his hips, and therefore will be able to tell exactly where the ball's going to go and get yourself in the right position, even though you can't see it.
Okay. Taking Ronaldo on in the seemingly impossible test is an amateur footballer called Ronaldo.
Come and join me, Ronald.
Ronaldo. So we've got Ronald in, same age as you, roughly the same height. We're going to use Ronald as a comparison to see how he reacts doing this same test.
Ronald is going first. So what does the night vision reveal? Ronald misses the ball by quite a way. Perhaps not that surprising. But can Ronaldo do any better?
How to do once. That's what's straight in the back.
The night vision replay shows how differently Ronaldo performs. To confirm that wasn't just luck, another attempt.
Love that. Because
And the replay?
Yes. That was fun.
how hard is that? It's difficult because it's changed a lot.
Yeah.
You're just focusing more on after no light. You have to try to. So this is the start of cognition.
Human reaction time is about 200 milliseconds, and by 500 milliseconds, Ronaldo's subconscious has interpreted Andy's body language, worked out which direction the ball will go in, calculated its speed and trajectory, and then programmed his body to reach it at the optimum moment.
It's almost as if they're doing maths in their head, even though they wouldn't be able to describe it to you. You could see from their performance that Ronaldo saw the first part of the ball flight information. He picked up information from Andy kicking the ball and was able to move his body into the correct position. This is because he's processed the information. He has experience of where the ball is likely to go. He's able to analyze all evidence that's coming towards him and therefore connect successfully with the ball. Ronald, on the other hand, doesn't have the same level of experience. He would have watched the ball flight. It's likely that he will just have had his eyes on the ball instead of picking up any information from Andy himself. And then when the lights go down and it's black, he's kind of stranded in no man's land a little bit and just has to do his best to react, but is naturally going to struggle with that.
So far, the lights have been shut off when the ball is in midair, but now the lights will be turned off just at the moment Andy kicks it. Can Ronaldo score with only Andy's body language to go on?
There you go. We'll get one. We'll get it.
Yeah, good.
Okay, Ronaldo.
Like,
Great finish. Great finish. That was a great finish.
Yeah, of course. I mean, I'm always for, this was the days that Ronaldo was playing for Real Madrid. And one of my colleagues, Javier Medina, he really was in favor of Madrid, but I, of course, was more for Barcelona. Even so, I'm showing his exceptional skills. My point is that I think this is a nice experiment to show transition from, let's say, direct sensory motor learning to what is the beginning range of what I feel is cognition and cognitive learning. Because there's the initial stimuli, and then for a whole period, maybe in this case, just one, few hundred milliseconds, but in that period, there's a lot of going on in his interaction between cerebellum and cerebral cortex. And we have replicated this in mouse experiments. So if you think about what I said about delay conditioning, during delay conditioning, you give a conditioned stimulus, and then during the whole period, we keep giving the conditioned stimulus, like a LED light or whatever. Now, in this trace conditioning experiments, we only for a very short period give a conditioned stimulus, and then for a long period, just like in the Ronaldo experiment, we don't give any stimulus. Yeah, and then we give the US basically at different intervals, sometimes in a narrow interval, always the same. Sometimes we spread it around to look at, while we are studying the different conditioned behaviors, like the timing thereof and the amplitude of the conditioned response. And basically what we see is indeed that you can train a mouse to do this. Of course, this takes a lot longer than normal sensory motor learning with delay conditioning. But if you take weeks of training, the animals do really well, and they can do it. And what you also see, it's not only suppression that is very dominant, but you have a much more heterogeneous kind of firing patterns, showing that multiple modules there start working at the same time. And we see this actually during all forms of cognitive learning. It's never either upbound or downbound. It's always a combination of those, no matter what kind of behavior you're studying, in mice or in monkeys or whatever. And you see this also here in this graph. You see the activities of the simple spikes going in both directions, both upbound and downbound. And then even within that range, there's a heterogeneous pattern, both among the upbound and downbound modules. And that's just like a return phenomenon. Okay, now I will show you a few other behaviors that are interesting for cognitive learning. This is a paper we did with Zeeuw in 2018. And here we use the discrimination learning task, a left-right task where the mouse have to first sense with his whiskers whether there's a rod in the left or the right field of his whiskers. And then during 1.3 seconds, there's nothing. Yeah, so there's no stimulus. This is when the real, let's say, cognition is supposed to take place. And he has to remember whether the clue was on the left or the right side. And then when the response can be initiated based upon a trigger, he has to choose based upon the initial cue whether he should lick to the left or the right side. During these activities also, you have an interplay of two different modules at least. One was the upbound module by which he could detect whether the whiskers was on the left or the right side. You need to protract the whiskers, which is done with the whisker system. This is an upbound module. And then in the end, the licking response and the interval between, let's say, the sampling stage and the response stage has to be mediated by a downbound module. So here too, they interact continuously together, and you can see it in the recordings of your simple spikes. So again, this is a kind of cognitive learning paradigm in which there's a combination of different modules. The same thing we did in another, not a left-right discrimination learning task, but a go-no-go task done by Lorenzo Bina and Lawrence Bosman and myself. And here the mouse again has to learn whether there's a rod in the front or in the posterior part of the whisker field. And based upon that, he can then get a water reward. He can start to lick and when the timing is right. So first he hears a sound, he's not allowed to do anything, but when he touches the rod in the whisker field, when it's in the right position, then he's allowed to drink. And not when it's in the posterior part, only when it's in the anterior part. And it takes him like, you know, about a week or so to learn this. But by the time he's learned it, you can see a very clear change, upbound changes and downbound changes in the different parts of the cerebellum, as well as even within single Purkinje cells. So what's really fascinating here is that initially the complex spikes go up and respond to sound, but later, when the mouse is an expert, the same Purkinje cell with the same complex spike response is not responding or hardly responding to the sound, but is responding to the touch. Yeah, and the simple spikes are driven by that. So they also move forward, meaning that you can trigger during the cognitive process, you can learn the mouse to learn basically a new sequence in activity and new sequence in the behavioral response. And we have been, you know, we wondered, of course, when we had these initial recordings, we could say, well, maybe these are different Purkinje cells or not. So we have actually proven that they can be of the same cells by recording at the complex spike activity in vivo of the mouse. So here you see
the sagittal tree of the Purkinje cell activity. You see calcium influxes. Yeah. We do this with the miniscope on top of the mouse of the head, so we can follow them during several weeks of training. You see the climbing fibers climbing into the dendritic tree, and they give this huge calcium influx, and you can follow them over time. So in this case, we followed the same cells for a period of 20 days, and you can see that initially the cells respond to the sound. At the end of the training, they respond not to the sound anymore, but to the touch, which is quite unexpected. It means something must be happening inside the inferior olive that we didn't understand before. So as a result, we started also look at the inferior olive itself. I mean, Jeremy and I, we were talking in the car about the principal olive. Well, what you see here, this is actually the principal olive, all of this, and this is a small accessory olive. And the principal olive gets a lot of input from the cerebral cortex via the meso-downspheric junction. But when you look at the cells in this part of the inferior olive, they look like this. Yeah, so they have a beautiful cell body, and then they have these dendrites that curl back. Yeah, and each dendrite has a numerous amount of spines with extremely long spine necks, longer than any part of any neuron in the brain. So there's, there's a huge amount of possibilities to collect information. And importantly, every little spine that you see of the inferior olive, when you look at them in the electron microscope, they have a gap junction. So they're connected to other olivary cells. And this means they can have all sorts of computational features that are not available in cells that have no gap junctions. We just published a paper in eLife and computational neurobiology, computational plus biology, to show this. So here you see a gap junction at high magnification, 7 layers showing the typical features of neuronal gap junctions with attachment plaques on the side. And the interesting thing is that you never see gap junctions between dendrites of the same cell. There's always of different cells. So here we intercellularly injected that cell, the olivary cell, indicated in black, and then in green you see the spines of another olivary cell, and only they are connected by gap junctions, but not within the same cell. They together form a glomerulus, so you have multiple spines in the core that are surrounded by chemical synapses. So in this case, you see the black dots, they indicate gold particles reflecting GABA, inhibitory neurotransmitter, and in blue, you see excitatory terminals. So in the same glomerulus, you have both inhibition and excitation to control the electrotonic coupling. Now, to show that this is really possible, we also combined actually anterograde tracing together with HRP and post-embedding gamma immunocytochemistry from the cerebellum nuclei and looked at the terminals in the inferior olive to see whether they indeed were closely terminating on-site the coupled gap junctions. You see that here. Here's a gap junction, and here you see a double label terminal with HRP from the cerebellum nuclei and post-embedding GABA strategically located synapsing onto these coupled spines. So this generated the whole idea that when these terminals are activated, you could get a shunting current here, thereby controlling the synergy, the synchrony between firing cells, Purkinje cells in the cerebellar cortex. So this experiment I did actually together with Rodolfo Linas, and we recorded from many cells in the cerebellar cortex while giving picotoxin, which blocks the GABAergic terminal. And you see what happened with the synchrony after you give picotoxin. So spontaneous activity, there's some synchrony, but when you block the GABAergic input, the leakage current is gone, and you get a huge synchrony. So you can really manipulate the set of cells, the network of cells that are firing together in the cerebellar cortex. So that was an important point. So you indeed are able to compose the ensemble of cells of the network through this mechanism. And the next question was, apart from this inhibition from the cerebellar nuclei, what about the projection from the meso-downspheric junction to the inferior olive? Are they excitatory indeed, as predicted? Yes, they are. You see them. So they have asymmetric synapses. They're excitatory. These are typical inhibitory synapses from the GABAergic cerebellum neurons. And then the question is indeed, so how do these two inputs interact? For this, we use optogenetics. So we use different optogenetic dyes to simultaneously manipulate them, both the inhibition and the excitation, and we can determine the timing between the two, how important that is. And just to make clear how important that is, here you see oscillating olivary cells. In blue, you see the impact from the MDJ, and in red, the impact from the cerebellum nuclei. And you see that the spikes only occur after a certain interval between inhibition and excitation, in this case 150 milliseconds. And there's optimal inhibition after 50 milliseconds. So what basically the conclusion is that the inhibition from the cerebellum nuclei to the olivary neurons, they determine the resetting, they reset the clock for the whole network, and then the excitation comes in to either give the perfect inhibition or the perfect excitation. And so this means this is a perfect functioning clock, if you want that. Much better than neurons in the thalamus or cerebral cortex or MDJ. This is what the inferior olif is perfectly designed for. And all neurons in the principal olif have this too. Right, so this is important, we feel. Moreover, in the principal olif, you get this huge input from the MDJ that in turn gets input from the cerebral cortex. So here you see a piece of the cerebral cortex labeled with transneural viruses. There's huge numbers of pyramidal cells that project to the MDJ and from there directly to the inferior olif. We made a whole map, and basically there's virtually no area of the cerebral cortex that does not project through the MDJ to the principal olif and the MAO. The DAO is different, the caudal MAO is also different, but the whole PO as well as the rostral MAO get huge input from cerebral cortex through the MDJ and a certain topographical relation. Also, when you then indeed manipulate the system, this is what we're doing now. When you manipulate the MDJ while you give this discrimination learn and go no-go task, as I described before, then this part doesn't work anymore. Everything behaves still like this, and you don't get the well-trained mice that you can do when the MDJ is intact. So this is what we're doing now in terms of trying to bring it all the way to cognition. We also do similar experiments in primates, where we really think there are so many tasks. I will also show you a task in the clinical part of my next part of my talk. But it's very clear that this whole projection from cerebral cortex to MDJ to principal olif is really important for cognitive learning, because there are cases where there really is no movement at all, as I will show you in the last part of the clinical test. So let me go back a little bit to why I think this is clinically relevant. As I said, in the Upbound modules, the baseline firing frequency of simple spikes of the Purkinje cells is around 50 hertz. In the Downbound modules, it's more like 90 hertz. So there's a clear difference, and it's a very robust difference, having all sorts of molecular causes. But it also makes it clear that the chance to get cytotoxicity in the Downbound module is much greater than that in the Upbound module. This is huge. You have to be aware this constantly while you guys are sitting here. Your downbound Purkinje cells are firing 90 hertz. And if you make a movement that requires it, it's even going up to 150 or higher. Even in your sleep, cells keep firing at a relatively high level. So these cells must be very strong to survive, for 100 years or so. Now, so when you plot all the mouse models that are available, I mean, there are many more than these, but I just listed this for now. And when you look at all sorts of cerebellar diseases, where you see where the Purkinje cells are dying when they are suffering from progressive ataxia, you see that on almost all cases, the Purkinje cells in the downbound zones, the cerebellum negative cells, they start to die first. Eventually, in some cases, like with COCNA1A mutants, that can also, other cells die as well, but they start
with the Zebrain negative cells, the downbound cells. Yeah, or what you see in certain diseases that the anterior lobe is most prominently affected. In all these cases, it's not like, you know, black and white, but it is by and large, maybe between 80 and 90%, you see these phenomena very prominently. There's one real exception in which the upbound cells, the Zebrain positive cells, start to die first, which is in the nervous mouse. This is early onset, not progressive. It's related to mitochondrial processing. Very interesting, but they have exactly the opposite pattern. I can show you the images. But given time, I will now focus on a set of experiments we did in Cognition mutant mice and the related patients, which are the SCA6 patients in this case. So here you see, for example, the distribution of Zebrain in a wild-type mouse, and here you see what's left of Purkinje cells with the general Purkinje cell marker in the leaner mouse. Yeah, so you see that only the Zebrain positive cells are sort of surviving. And this was already done by many others, by Hegroth and Abbott a long time ago. And I can show similar patterns for Niemann Peak mice and many others. When you look at the survival of cells, of Purkinje cells, the Purkinje cells that are positive for Zebrain 2, they are the one who survive longest. Now, the Cognition mutant, as the leaner and the tottering are, you can also find it in patients. The SCA6 patients, they have progressive ataxia, usually occurs between age of 40 and 50. And what you see there too is that the degeneration occurs predominantly, not exclusively, but predominantly in the anterior lobe. Just look at here, lobule 6, yeah, over control subject, 70 years old. And here, a person suffering from SCA6, 65 years old. Look at the difference in size of the anterior lobe. And this is usually the pattern. So what we have been doing, and again, just want to remind you that the number of Zebrain negative cells in the anterior lobe, yeah, is much, much greater than the number of Zebrain negative cells in the posterior lobe, or here, for example, in the vestibular cerebellum. So there's this correlation between the presence of Zebrain positive and negative cells and the different lobules is not like 100%, but there is a clear trend. Now, what we did in these patients with SCA6 as well as control subject is the following experiment. We call it weight discrimination task. We published this in Brain in 2019. We made movies, and we asked first the normal subjects to analyze the movies. It was explained to them that in all the movies that we have, the people who were subject of the movie, presented in the movie, had to carry a weight of a certain relatively little water, a lot of water, or something in between. And so there were 3 different weights. And in the movie itself, if you analyze the kinematics of the movie, there was, of course, a different change in kinematics of the arm movement when you have something with a low weight or a heavy weight. And then on top of that, we had the situation when there was no sleeve around the arm, which means that you do not only see the kinematics, but you can also see maybe some shape of the muscle activity. Yeah, and we had a condition where there was a sleeve on top of the arm. So here you only had the kinematics. Here you have both shape and kinematics. And then we first looked at the fMRI activity. And again, you could see a very clear activity in lobule 6 in all cases. You can make the the comparisons with sleeve and no sleeve both, or subtract them. But in all cases, you found activity in which you could really show that activity in lobule 6 is related to detection of the kinematics. Then when we subjected SCA6 patients in which lobule 6 was not prominently present anymore, you could see that there was a clear significant difference in the weight discrimination task, both actually with sleeves and without sleeves. So they clearly have problems identifying the kinematics. And to be sure, I mean, these patients did not have to make any movement. They were just judging
as a cognitive task what was going on the screen. Of course, you can say, well, maybe they made different eye movements or things like that. We have tested for that. There were no correlations. And so again, we think it shows that the cerebellum is really important for cognition. Okay, so with this, I would like to conclude my talk. I hope I convince you that there are upbound and downbound modules that have a very clear role doing sensory motor learning. Doing cognitive learning, you have a synergy of multiple modules operating at the same time. It's a more complicated task, and they do have clinical implications. This work was done together with many people, most of which are listed here. I would like to thank a few. For example, Abdel, he was instrumental in the study I just talked about for the SCA6 patients, as well as Robin Broerse. Henk-Jan Boele and Michiel ten Brink were important for eyeblink conditioning. Gao and Noah Lee were important together with Karel Svoboda for the task of discrimination task. And the VOR studies were mostly done by Kai Vogts and Martijn Schoonewille, and the trace conditioning by Dave Kammerheim. Thank you.